Hello everyone, my name is Saram Kilji. I am a biology major here in the College of Life Sciences, focusing specifically on the discipline of bioinformatics. Today, I'm excited to share a little bit about my project which focuses on the evolution of certain genes called NLRs in a Hawaiian flowering tree known as Metrosideros. And so what I have here ready for you today is just really an exploratory data analysis with a handful of preliminary results. My project is still very much in its infancy, but I'm still excited to share what I've been able to accomplish so far. And so, what exactly are NLR genes? Since many plants do not have the same specific adaptive immune systems that us humans and other animals do, plants have to rely much more on broad immune system defense mechanisms in order to defend against pathogens. One of these mechanisms are called nucleotide-binding leucine-rich repeats, or NLRs. These NLR proteins are important key players when it comes to many plants' abilities to ward off infection. And while today I'll be discussing their evolution within the Myrtaceae family, study of NLR genes is broadly important to understand due to the fact that NLRs are present in many of the crops we grow and the plants that maintain our ecosystems. And so one of the many ways we can glean more information about NLRs is by doing a comparative analysis of the genes that encode NLR proteins. This comparison of NLR genes both within species and among species can help us to understand both the conservation and variation of NLRs among plants. And so one of the things we wanted to look at was whether the variations of Metrosideros trees that had these more hairy, rugged, and generally tougher leaves would also translate that physical toughness chemically into the NLR genes themselves, in comparison to those of Metrosideros plants that had those more naked leaves. And when I say toughness, what I really mean by that is that these leaves from these plants could withstand more harsh physical and environmental conditions. Initially, however, we started with the hypothesis that the naked taxa would confer a greater number as well as more diverse NLR genes. And so what you see here on the side in this picture below is just some of those leaf samples. The ones labeled I, C, F, and R are the ones that came from those tougher, hairy plants, whereas the rest came from those more naked plants. Another thing to quickly know before we move on to the methods is that in addition to using Metrosideros, we also use another tree species called Eucalyptus grandis in this comparison in order to look at how NLR genes not only compared among Metrosideros, but also among different species in the Myrtaceae family overall, which this family includes both Metrosideros and Eucalyptus. And so one of the main ways that we can do a comparative analysis is by drawing attention and getting the parts of the DNA sequences we want, which in this case will be the parts that code for NLR proteins. Bioinformatic annotation tools allow us to essentially highlight and pull out these desired segments of the DNA. And so here in this picture, I just really wanted to draw attention to a quick example regarding how I think about annotation, which is really just highlighting the parts of the genome that we really care about and taking them out and extracting them. This specific annotation tool here, which is available for public use, is specific for annotating NLR genes. And so what you see here is really a figure that shows a more in-depth algorithm regarding how the genome is positioned, parsed through in order to find the genes they were looking for. Steps B, C, and D specifically here simplify this process. Again, this process is a little bit dense, but all you really need to know in the end is that we're pulling out those NLR genes out of the genome so that we can look at them on their own without the clutter of everything else. And so the result of the annotation process can be seen here in an example for that of Eucalyptus grandis. And so to just explain this a little, the first column here is really just represents the name of each gene. The second one here tells us whether a gene is complete or partial, which correlates to whether the NLR contains everything that actually makes up an NLR, or only parts of it make up an NLR, hence partial and complete. And so doing a little data analysis on that file that you saw before, for both Metrosideros and Eucalyptus, we can glean some information in regards to the number of NLR genes in each taxa. The fact that the pubescent variation has almost 100 more NLR genes than that of the naked one does seem to support a hypothesis that the pubescent variations of Metrosideros are tougher, chemically speaking, than those of the naked ones, which again are thought to be less tough. Another data manipulation allowed us to look at which chromosomes these NLRs are coming from. While there isn't much we can really we're able to really gather from this on its own, combined with an analysis I'll talk about here in a little bit toward the end, we can hopefully see whether the chromosomes these NLR genes come from confer any sort of significance. So what I did next here was take each of those NLR sequences from Metrosideros and from Eucalyptus and essentially line them up sequence by sequence by sequence. Then using a combination of the NLR annotator tool, which I just mentioned before, as well as a program called AliView, perform something called a multiple sequence alignment. This step will not only help us to examine characteristics, but it's also the input that we will need in order to generate something called a phylogenetic tree, which I will discuss here just shortly. And so here's what that looks like. Each NLR sequence we pull out of each of the genomes can be seen here on the left side with its name. And to the right of each name, we can see the protein sequences that corresponds to it. And so essentially what the algorithm has done here is like each of the sequences to give us a better visualization of where the sequences are similar and where the sequences are different. This visual comparison, along with other qualitative data provided by the solver, can help us understand which parts of NLR proteins have been kept the same across the different taxa, which parts are different. In this specific image of the comparison, we can see that there's a lot of variation in the protein sequence, potentially indicating that the NLR genes are not very well conserved across these taxa. We can get an even better look at how these sequences compare by doing a phylogenetic analysis. This analysis will create a phylogenetic tree that we can look at in order to examine the relationship among taxa. Here, we use a program called Raxamol that takes in the multiple sequence alignment as an input that we just talked about before in the previous slide, and outputs it as a file that can be used to generate a tree. That file that Raxamol generated can be inputted into another software called ITOL, which is able to visualize that software in a graphical way that allows us to interact with it and see it in a more eye-pleasing kind of way, which I'm about to show you guys next. And so here again, this is what that output looks like with everything color-coded as well. The red here represents Metrovar incana, the pubescent taxa, the blue represents Metrovar glabrous, the naked taxa, and the green represents the other tree species in that of Eucalyptus grandis. This tree shows us the relationship between these taxa, and looking at it a little more closely, there are some interesting things to take note of. This part right here in the top left-hand corner within the bracket is quite dense with the two Metrosideros taxa. You have to go quite a bit of ways before you start running into any major occurrences of the Eucalyptus NLR genes. Overall, to me, this indicates that while there's a lot of similarities between the two species, certain NLR genes may have diversified more so in Metrosideros, while others may have diversified more in Eucalyptus. One thing I would potentially like to look at in the future and do in a future analysis is add more Metrosideros taxa to this analysis to get a better idea of what's going on overall. Another thing we could potentially look at is gene order. NLR genes, as I mentioned before, can be mapped to certain chromosomes. Looking at the order of where and when these genes can occur can provide insight into whether NLR protein mechanisms could potentially be dependent on gene clustering or gene regulation. This process of looking at conserved gene order or lack thereof is commonly referred to as synteny analysis. Using the SynNet pipeline here, we hope to find information in a future analysis regarding the synteny of NLR genes in Myrtaceae and the broader Myrtaceae family. And so the conclusion and the key takeaways that I hope everybody takes away from this is that NLR proteins are really important key players when it comes to plant immune defense response, since they don't have a lot of the stuff that we humans and animals have to fight against pathogens. And by doing comparative analysis of NLR genes among plant taxa, we hope to better understand their evolution as well as their potential mechanisms and how they work within the plant cells. And I know while all this information is very preliminary, early results do indicate or may indicate that there is significant NLR evolutions in the Hawaiian Metrosideros, and that pubescent variations of Metrosideros polymorpha may confer a stronger NLR response than when compared to those of their naked counterparts. And again, I'd like to thank the undergraduate research program as well as the office for allowing me the opportunity to present this information in this project. I also like to give a very special thanks to Dr. Stacy and Dr. Choi, who are my two, Dr. Stacy from UNLV and Dr. Choi from NYU, for providing me a lot of help with this project. Thank you so much.